It is with great pleasure that I present our second keynote speaker of the day, Professor Jim Leebens Mack. He's a distinguished research professor at the University of Georgia, where he has been since 2006. He received a bachelor and a master's degree in ecology and evolutionary biology from the University of Minnesota, and his doctoral degree in botany from the University of Texas. His lab uses phylogenomic and comparative genomic approaches to explore the ecological, genomic, molecular, and developmental processes that contribute to phenotypic diversification and speciation across the green tree of life. Jim has a particular fondness on monocots. Welcome, Jim, and thanks for being here with us for this second keynote talk.
Oh, thank you, Adam. Well, thanks so much to the organizers, and it's wonderful to be here in San Jose. So, and thanks to Tom for giving us a great history of one side of the phylogenomics coin. I'm going to talk a little bit more about that and then flip over to the other side. I'll show you what I'm talking about here. So everyone's familiar with this term. I think in this audience, I think most people would be sign on to the second term that I think was coined independently by multiple systematists. Nothing in evolution makes sense except for the light of phylogeny. The population geneticists, they'll argue with that a little bit. They'll say population genetics. I think both are true. We jump ahead today, so these are the things that made sense to Charles Darwin when he was late 20s, early 30s, and touring around the world and sketching in his notebook, seeing phenotypic variation and wondering about how much of this is due to shared ancestry versus evolution after speciation. Jump ahead now to the last 10, 15 years. We're in the age of phylogenomics, and this is a two-sided coin, as I mentioned. On the one hand, we do the thing that Tom was talking a lot about this morning, and I've heard a lot about in the Mike heterotroph session, using genome scale data to resolve phylogenetic relationships. On the other side of that coin, once we have an understanding of phylogenetic relationships, then we can ask questions about gene family and organism and genome evolution in the context of our organismal phylogeny. So I'm going to be talking about asparagus today, and this work is following off of a paper that was published in 2015, but actually it was a paper that I was talking with Maria Norup and Ulla Seberg and Gita Peterson and others at the MONOCOT meetings that occurred in Copenhagen years back. And following that meeting, Maria led the charge as part of her dissertation work to make a phylogeny, a pretty comprehensive phylogeny for asparagus, and identified that there seemed to be two origins of Dioecy within the genus. And here's the phylogenetic distribution. So asparagus, its cradle of diversity is in southern Africa, and but as we'll talk about, Maria talked about in this paper, but we'll talk more about where we're at with this today. There was range expansion out of southern Africa into Eurasia and then the Mediterranean Basin, and those range expansions were associated with shifts to Dioecy. So that paper was based on clastome regions and one nuclear genome. In order to refine our understanding of relationships, we've developed a bait set for the Asparagus. Ooh, and I feel your pain. I remember you were doing this morning, so I'll try not to do that too much. But so we've developed this Asparagus 1726 data set. Always got the number there. This is described in a paper that is in press in Apps. But if anyone is working on Asparagaceae and wants to use a bait set, the baits are available through Arbor. And if you feel free to contact me if you have questions about this. We developed this bait set because we had genome sequence for Asparagus, officinalis, and a few yucca species. And we then identified 1726 pure single copy genes in those two genomes. That included 200 and some odd genes that are in the 353 set. And I think it's as we're developing these bait sets for particular groups, going back to the 353 set and making sure that some of those genes are in that set, I think is going to be a useful thing. So we're able to weave all these phylogenomic data together. But that bait set then, in this paper, we've applied that to a sampling of the subfamilies in the Asparagaceae, Gavoideae, Asparagaceae, or Asparagus is shown here, but then we had these other members of representatives of the various subfamilies. Here we see our capture efficiency highest in the taxa where we developed the baits with it, where we had the genomes developed baits, but still over 1,500 genes that we're capturing pretty faithfully across the family. So using that bait set, and really by a lot of this I'm saying that the development of that bait set was really led by a PhD student, Phil Bentz. I really wish that he was here talking about this work, but he's got a postdoc lined up, and he's working hard to wrap up his dissertation in the next couple of months, and so he didn't have time to come to the meetings, unfortunately. But Phil, with help from Sandy and John Burrows, a couple of ecologists and taxonomists, asparagus taxonomists in Southern Africa, we were able to sample across the Asparagaceae. And much like Maria found, we see, as I'll talk about, two origins of dioecy, it seems, within, or two dioecious clades within this Asparagus clade in the family. And that's what I'm going to be talking about. So that is the first side of the coin, and I'll come back to it, of the phylogenomics coin. Using genome scale data to reconstruct relationships, the second part is using that understanding of relationships to understand genome evolution. In particular, I'll be focused on a sex chromosome evolution. So this whole process, it really starts with diverse sampling of specimens from various environments and good phenotyping of those specimens. So biodiversity science is critical to both sides of the phylogenomics coin, obviously. What we're working on now is a distributable pipeline that would allow us to take genome data from whatever taxon, combine it with genomes from databases like Plaza, somebody showed slides on this morning, or a phytosome or NCBI, and then take those genomes, circumscribe gene families, make multi-sequence alignments, gene trees, reevaluate those gene trees in the context of the species relationships, and synteny data to annotate and perhaps refine those gene trees. And then this organization of the data is very useful for molecular biologists and all of biology to better understand the biodiversity that we find in each one of the plant genomes that we're studying. So if we want to really understand the genetic basis of some of that phenotypic variation, One thing, the most powerful approach is to compare the genomes of closely related organisms, or in the case I'll talk about today, in some instances, two genotypes within the same species, or multiple genotypes within the same species, to make that kind of do a GWAS analysis or a genome-wide association analysis to identify the parts of the genome that are associated with the phenotypic variation of interest. Dioecy is a trait that has evolved multiple times throughout the flowering plant phylogeny. Sex chromosomes and dioecy and sex chromosomes have been identified within across about 50% of angiosperm families. We see here the number of species that are dioecious, and in some of these families, we have multiple origins. Often we have multiple origins within a family of dioecious species. So we've got hundreds of origins of dioecy across the flowering plant tree of life. And this is, really a powerful data set for allowing us to ask, well, what are the sorts of things that might be pressures that might be driving the origin of dioecy? What are there some general rules that we can ascertain for the way in which sex chromosomes are evolving? In mammals, placental mammals, there's one origin of sex chromosomes, and that happened a long time ago. There's been a lot of hypotheses about how these sex chromosomes have evolved, but they're really hard. It's hard to test those hypotheses because the script of evolution has been overwritten over that 150 million years. It's the last common ancestor of all mammals that, including us, that have XY chromosomes, X and Y chromosomes. In flowering plants, we've got a very different situation where we've got sex chromosomes, dioecy, and sex chromosomes evolving in some instances maybe 80, 90 million years ago, for instance, in the Salicaceae, or and in other instances, just a few million years ago, as in Asparagus, as I'll talk about. So this is a paper, if you're interested in this topic, lead author Sarah Carey, Alex Harkis at Hudson Alpha. So one of the big questions is, well, with the origin of sex chromosomes, is it one gene that's responsible for determining whether an individual is going to be male or female, or are there multiple genes that are contributing to the differences between males and females? And Brian and Deborah Charlesworth were big proponents of this idea that maybe there may be two genes in many instances that might be responsible for the origin of a sex chromosome that are kind of having antagonistic effects. So in an XY system, we might have a gene that suppresses femaleness or female function associated with a gene that's necessary for male function. And the Y chromosome will have both of those genes. So you have the male phenotype. The X chromosome doesn't have either of those genes. So individuals that are XX will have the female phenotype. Or do we have a situation like this where there's just one master regulator? And this is something that I think folks that are around the world that are studying sex chromosome evolution are, as I'll talk about, really curious to know, well, is one of these two situations more common than the other? Under what circumstances you have a two-gene or multi-gene system versus a one-gene system? So I'm going to talk about, as the title of my talk suggests, asparagus, and we started with this. This was started as a collaborative project, but Alex was a student in my lab, and so he was really kind of leading the charge from my lab. But we're also involved with folks from the Netherlands and China and Italy, as I'll talk about. We started off by sequencing an individual that had a YY genotype. So in asparagus, you could have you can have these super males that are YY. Actually, these genotypes are very important in the asparagus breeding system. So we started off by sequencing one of those individuals, and then identified the regions in a portion of that chromosome that was not on the X. There was just a few genes that had decent annotations there. And I'll give you the punchline. The genes that are on either end of this region of the Y are the sex determination genes. This gene it was a domain of unknown function gene, which is kind of scary. We still have no idea what the molecular mechanism for its action is, but it'll talk about when you knock that gene out, you convert a male to a hermaphrodite. So we call this gene suppressor of female function. On the other end, we have this TDF1. It's a MIB transcription factor that has been, as I'll show, shown in Arabidopsis, a homolog has been shown in Arabidopsis to be necessary for full anther and pollen development. So how did we get? We saw that there were these genes in this segment that was specific to the Y chromosome. How do we figure out the function? And we used, in collaboration with our folks at this LIM group, this breeding group in the Netherlands, they were able to make a bag full of XY male seed. They did this by crossing a YY individual with an XX individual. All the seed was then XY, irradiated that seed, and then planted out tens of thousands of individuals in a field in Peru, and then went around and looked to see, well, which of these individuals are producing fruit. And so I'll start by talking about one individual that was producing fruit. When we looked at the flowers, we saw that they were hermaphroditic flowers, and we resequenced that to figure out, well, what was going on with that individual. So we resequenced, and it had a deletion for this domain of unknown function gene that we had identified was in the Y-specific region. And so that, that was a big day in the lab for Alex. He was the one that was doing the read mapping and the analyses. I think we got it. We've got this gene that's in the sex termination region that has a deletion of one of its exons, and that's our hermaphroditic phenotype. We went back and we're talking with our Italian collaborators about this. This is Agostina is a asparagus breeder in Italy, and he said, you know what, I have got this population that is segregating for hermaphroditic and male individuals. So let's go back and resequence some of those, the hermaphrodites and males, and see what the difference is in their Y chromosome. And it turned out that there was a one base deletion that resulted in a nonsense mutation in the same exon that was deleted in our gamma mutant. So that was, again, another great day in the lab. We're like, okay, we've got two mutants that are all pointing the same gene as the one that's responsible for suppression of female function. Phil, more recently, has gathered sequences for a number of dioecious species, and resequencing these members of these different these species and mapping reads back to the Y chromosome, he was able to, and the rest of the chromosomes, was able to construct full assemblies for two copies of this gene, one autosomal and one that was only found in the males. And then a gene tree for that showed that there was that all these male copies form a clade. So in the last common ancestor of Cochin chinensis, Taliensis, Cusiensis, and officinalis, there apparently was a gene duplication event, and one of those copies, neofunctionalization of this copy, taking on the function of suppressing female development. Cetaceous was our outgroup, genes were an outgroup for this. It is a hermaphrodite. All right, now what about TDF1 I mentioned on the other side of this chromosome, this MIB transcription factor. So as I said, in Arabidopsis, it has been shown to, knockouts of this gene have been shown to actually have a phenotype that's similar to the female phenotype in Asparagus. So it initiates both male and female parts, initiates the stamen, but late in development, the stamen starts degenerating and pollen is not produced, very much like we see in female asparagus flowers. So that made sense. But then we wanted to follow up by doing some EMS mutagenesis in this case, so chemical mutagenesis. This is work done by Bart, again, in the Netherlands with our collaborators at this Limb Group, this breeding company. And so mutated a male individual and found that he had some neuters, and went back to look at, well, what's going on with the TDF1 gene for those neuters, and in fact there was again a nonsense frameshift mutation supporting the function of TDF1 is the same function that we see in Arabidopsis. So here's the bottom line. We can have XY male if we mutinize, and there's a deletion of the entire sex determination region. So we had some of those as well. We had 2 of those in that initial experiment. And in that case, we convert a male genotype to a female genotype. If we just delete SOF, then we convert a male to a hermaphrodite. And if we just remove TDF1, we convert the male to neuter. So I mentioned before that Phil was resequencing to a depth of coverage of somewhere between 30 and 15x coverage, a bunch of other species of asparagus, and then map those reads back to our Asparagus officinalis genome. And what he noticed was that here we've got a reference genome, Asparagus officinalis. Meridimus, close relative, has all of the same genes. Had reads that were male-specific, mapping to all the genes that were in the officinalis Y chromosome. Brachyophyllus oligoclonus, they had most, but not these 2 genes, and so on. As we go back down the tree, we see that we have the content of the gene content that we found in the Y chromosome in officinalis is we're basically accumulating from this last common ancestor of Cochinchenensis and Taliensis. This is where that duplication occurred. We're accumulating over time the genes that we've characterized in the full assembly that we have for officinalis. Interestingly, Cochinchenensis and Taliensis, they have this male-specific copy of this domain of unknown function gene, or this suppression of female function gene, but they don't have this TDF1 as male-specific. TDF1 is autosomal in these 2 species and the clade that these 2 species represent. And we're still in the process of trying to figure out, well, what's the male promoter in this clade? And also, is TDF1, we know that it is necessary to for development of the full development of pollen in males, but is it sufficient? We're working on addressing that question as well. So just what about this other group up here? We've got this other what Maria found as a separate origin of Dioecy. We've, in our analyses, found better support, even better support for this as a separate origin. And it turns out that these two species represent a clade that is restricted to the Mediterranean Basin. So we've called it the Mediterranean Basin clade. Something else that's kind of interesting about this clade is that we do have separate sexes. We have some plants that produce fruit and some plants that don't. But the males actually, they do produce pistils that look pretty much like the females. It's just that those anthers are, I'm sorry, the stigma are not functional. Pollen is not germinating on those stigma and affecting pollination. So that might give us hints about what might be going on there. This other clade that includes officinalis and Cochinchinensis italiensis, the ones without the suppressor, that don't, without TDF1, they are part of this clade that is centered in Asia. Here's a plastome phylogenetic analysis. This was recently published in American Journal of Botany. One thing that was interesting about this analysis is that we identified that actually that best supported hypothesis for the relationships of these clades here. One, this is a hermaphroditic clade. Two, this is the Mediterranean Basin dioecious clade. And three, this is the Eurasian dioecious clade. The best supported hypothesis is that this is a polytomy. And I think that, that if we go back and look at our plastome analyses, that there'll be instances where it's not that we haven't collected enough data, it's that the true set of relationships is that there's a polytomy at that point in the tree. We know that there's variation in extant species that where we can if we were to sample from that extant species, we'd see that there's more than two lineages that might be coming off of a gene network within that extant species. So anyway, this is something that I'd encourage folks that are doing plastome phylogenomics to look at, testing for whether or not your best hypothesis, your best supported hypothesis, is that you have a polytomy on that point. The other thing I'll point out from this tree is that we put some dates on the last common ancestor of all the dioecious species in these two clades, and I would add this hermaphroditic clade here. This is work that Phil led, but done in collaboration with folks in Yunnan province in China. So using the asparagus bait set that I mentioned before, and before I showed you the kind of big picture of what the variation, what the phylogeny was for the subgenera within the genus. Here I'm focusing on the asparagus clade within the genus, and here's what we're seeing. We've got very now excellent support at 100 posterior probabilities of 1 at every node here. The last common ancestor of all members of this clade was hermaphrodite, and these are species that are native to southern Africa. And then again, at some point we've got range expansion, maybe 2 range expansions. One here that where the center leading to diversification in what is now Western China, and another diversification here in the Mediterranean Basin, or perhaps the Arabian Peninsula. Sister to that clade is this hermaphroditic clade, and these are mostly native to South and Central Africa, although some of them do get up into North Africa as well. So, okay, so that's the pattern. It seems like we have 2 dioecious clades what's going on within each of them. Well, and what's going on with that one clade I'd mentioned before that I'll show it here. This clade, including Cochinchonensis and Teliensis, these guys don't have TDF1. TDF1 becomes sex-linked at this point in the tree. So what's going on with them? So again, in collaboration with Zhengji Liu and Zaichao Mao in Yunnan province, we were doing some genome sequencing for two of these species, Teliensis and Cochinchonensis. And as we were gathering material to do that, we noticed that in populations, there are hermaphrodites. So most individuals are either male or female, but you find some individuals that are hermaphrodite. And actually, some of the folks that are analyzing that, growing these as a species for Chinese medicine, they're harvesting roots for Chinese medicine, they are actually preferring the hermaphrodites. Now, one thing that we found is that the hermaphrodites and XX females, they do have a copy, the autosomal copy of the STUF247 gene, but they don't have the male-specific copy, and this is those are restricted to the XY males. And so the male-specific copy is represented by this half coverage in males because there's XY males. So these reads are coming from the Y chromosome. These reads up here that don't have these gaps in that male Y-specific copy are from the X chromosome. So here's our hypothesis for how this is working. XY males have soft suppressing ovary development, but there's a Y-linked male promoter or repressor of a male repressing gene, which is unknown. XX females don't have soft nor this unknown male promoter, and XX hermaphrodites are missing soft, but they must have the functional male promoter or repressor of a male repressing gene. So what about the Mediterranean clade? Well, when we go back to the shotgun sequence data, we noticed that for males and females, so we looked at males, we identified chamers that were specific to males. So sequences that were only found, these are like 31 base sequences or stretches that were only found in males. And we mapped those back to the officinalis genome and found that those were all mapping back to an autosome, not to the Y chromosome. So it seems like there's a different region, portion of the genome that's responsible for sex determination. We go and we've now sequenced and using PacBio sequencing and Hi-C sequence. We've now the chromosomal assembly, actually the haplotype resolved chromosomal assembly for horridus. And we see on that when we map those the chamers and reads from males and females back to this one of these chromosomes, we find that there's this 10 megabase region that seems to be specific to the males. It turns out that the difference between the Y chromosome and the X chromosome in this region is that there's been an inversion. And following that inversion on the Y copy, this region has expanded. We've got a few more genes than we see in the X in this region, and a lot more repetitive sequence, as we might expect it to see, actually. All right, so I'm going to end now talking a little bit about our hypothesis about range expansion and two origins of Dioecy and Asparagus. So again, here we have our tree for the Asparagus clade with these two dioecious clades. The center of diversity is down here in Southern Africa. There is actually quite a bit of diversification occurring among hermaphroditic species in Southern Africa. But then we have what we're estimating is two separate expansions out of southern Africa, one to what is now Western China, the other to the Arabian Peninsula and the Mediterranean Basin. And these two range expansions are associated with the separate origins of Dioecy. Very quickly, that model is based on a GeoBears analysis, and I'll have to say that from that analysis, our likelihoods, our relative likelihoods for different ancestral positions on the map are not really high, but they are the highest. So here we've got all of the regions here are in the mix. We've got samples from across the asparagus phylogeny from all of those regions. And when we use this approach to model the ancestral state reconstruction, we can say here, this whole group, definitely its center of diversity is in Western China, but these others here, we're in the Mediterranean Basin or Arabian Peninsula, and both of those areas seem to have been derived from ancestral populations that were in South and Central Africa. And so, again, this is the summary of that, or the interpretation of the BioGeoBears analysis. It kind of makes sense that we have the origin of Dioecy associated with range expansion. We expect that Dioecy would be a way to prevent inbreeding in or selfing in small populations as these ranges are expansion. But we can't throw out the alternative idea that's been in the literature that sex-specific resource allocation is also playing a role. I'll leave it there, and thank you very much for your attention. Oh. Be happy to take any questions.
All right, thank you so much. Tatiana, you?
So I'm assuming that these are transcription factors, these sex determination genes that you found.
One of them is, though, the TDF1 is a transcription factor that's influencing the genes that are important in the development of the tapetum and pollen. The other gene is a domain of unknown function gene. We have no idea. The suppressor of female function, we have no idea what its function is. No one else does either. It's been implicated as being involved in self-incompatibility. So it's part of S gene, S loci in grasses. But even there as well, they don't know exactly what the molecular function is.
My second question would be like, do you know the relationship between these genes and the expression of other genes like the genes in the ABC model?
Yeah, so the ABC genes are all expressed in the way you expect. So in asparagus, they're initiating, both males and females are initiating both male and female parts. It's later in development that we have cessation of male development in female flowers or female organs in male flowers. So the ABC genes aren't part of the story at all. I don't know that they've been really implicated in any of the dioecious systems yet. I think in most systems, there's only been a handful for which we've identified the genes really responsible for sex termination. And in those instances, they've been genes that have been influencing sex termination later in flower development.
Any other question?
Sean?
So there are many other origins of dioecy. How does this system compare to what we know in other systems, and what do you predict about other systems?
Thanks for this opportunity to talk about this slide I glossed over before. So we're still trying to figure out, well, what are the common themes with the origin of sex chromosomes? Dioecy is a whole other ball of wax. But for sex chromosomes, again, there's only been a half dozen or so instances where we've identified the genes responsible for sex formation in dioecious species. And many of them are in the Salicaceae, poplars and willows. One of the things that we might have thought was that, well, and had been hypothesized early on, is that the size of this male-specific region in XY system or the female-specific region in the ZW system would be increasing over time to a point, and then as there's too much, a lot of repetitive stuff in the heterogametic sexes specific chromosome, then we lose that material. That's what's been hypothesized for R or Y chromosome. In fact, we don't see much of a relationship between the age of the sex determination region or sex chromosome and the fraction of the sex chromosome that is co-segregating with sex. And so I just showed officinalis and horridus. Horridus is a little bit younger than officinalis' sex chromosome, but we see there's great differences in the proportion of their sex chromosome that have the male-specific region, or sex determining region. One thing that we can say is that in all the instances we've looked at, gene duplication has been part of the story. So there's been gene duplication and one of those copies taking on some function in sex determination.
All right. Sorry, no more time for questions, Raymond. Maybe during the coffee, is that okay? Yeah.
All right. Thank you very much.
Thanks so much.